In this segment, we follow up on the last couple of segments about nucleic acids, where in the last two segments, we started by looking at the nucleotides as the building blocks of nucleic acids, such as DNA and RNA. And then from there, in the last video, we looked at how nucleotides bond to one another via those 3,5 phosphodiester bonds to yield strands of nucleotides, also known as our nucleic acids, which include DNA and RNA. What we are going to do in this segment is look at further levels of organization of our nucleotide strands to assemble into their functional units, into their three-dimensional structures that will allow them to act as the genetic repositories and the genetic material transporters around a cell. So in order to do that, we will go ahead and take a look at our whiteboard here. And our nucleotides, remember, up in the upper left-hand corner, our building blocks. Those are the structures that are shown in the lower right-hand corner here. That's our nucleotide. We link those together by joining the oxygen atom at the 3 position here with the phosphate group from the next nucleotide in the chain to make a strand of nucleotide that is up to millions of nucleotides in length. And we can abbreviate those nucleotides based on what is unique about each of them. And what's unique about each of them in DNA is the composition of the base. So the bases that we have are A, T, G, and C. Those are the ones that are found in DNA. And so if we are listing out the sequence of DNA, we can list it using those one-letter codes. The one-letter codes indicate that a nucleotide containing that particular purine or pyrimidine base is present. And so this strand of nucleotides represents the primary structure of a nucleic acid such as DNA or RNA. So an example of this primary structure, if we listed ATT, GA, CG, TA, et cetera, et cetera. I'm just listing out the names as a one-letter code of the bases, adenine, guanine, thymine, and cytosine, that comprise these nucleotide building blocks. So we would indicate and we would recognize based on this sequence that each of these are linked as we go down the chain via a 3,5 phosphodiester linkage. By convention, the way that this primary structure, the sequence of the DNA or RNA is listed, is that the 5' end of the chain is the end that has a free phosphate group, meaning it's the beginning of the chain where the 3' hydroxy group was the component that was reacting, and that leaves the phosphate group free, as we see in the lower left-hand corner. And then as we continue down the chain, at the other end, you would have a free hydroxy group at the 3' position. And so I'll go ahead and write that down for summary here. When we're thinking about the primary structure, the sequence of a nucleic acid chain, the way that we write that out by convention is that the so-called 5' end of the chain is listed first. And that's going to be the end of the chain that has a free phosphate group. On the other hand, when we get to the other end, the very other end, which could be millions of nucleotides away of this covalently linked chain, the other end of the chain is going to have be called the 3' end, and that is listed last at the far right. And that's going to have a free 3' hydroxy group. And we refer to this entire chain as the primary structure of the DNA or the RNA, the primary structure of that nucleic acid, what is linked all the way through by those phosphodiester bonds. This is analogous to the primary structure of a protein, where remember that the primary structure of a protein was just the amino acid sequence. Similarly, primary structure of a nucleic acid, such as DNA or RNA, is the sequence of nucleotides that are found there. And just like with the proteins, we could list the sequence in their one-letter abbreviations from N-terminus to C-terminus. Here, we can similarly do that, but we're listing from the 5' end to the 3' end. Also analogously to proteins, we can assemble these strands into higher levels of organization. After the primary structure, which is just the sequence of nucleotides that are covalently bonded together, we have the secondary structure. Now, if you think back to the secondary structure of a protein, the secondary structure of a protein corresponded to things like alpha helices and beta sheets, where the individual amino acids were engaging in hydrogen bonds and other interactions, electrostatic interactions with other amino acids to fold the protein or start to fold the protein up into these alpha helices and beta sheets, some areas of local organization. Similarly, with DNA, the secondary structure is also driven primarily by hydrogen bonds. And it's the secondary structure that we especially focus on when we're thinking about DNA, because it's the secondary structure where we get the classic DNA double helix. So that double helix is driven by hydrogen bonds. Specifically, to form that double helix, we assemble hydrogen bonds between purines and pyrimidines. So if we look in the upper right-hand corner of the slide here, we have our purines, which are adenine and guanine, and our pyrimidines, which are thymine and cytosine. Those are the bases, and those are what are going to participate in the hydrogen bonding to create the double helix. So specifically, the hydrogen bonds that we will obtain are between AT, so specifically between the adenine purine and the thymine pyrimidine base, and then GC. So these are our specific hydrogen bonds that allow us to form ultimately the double helix by forming a whole network of these across millions and millions of nucleotides. So the way that it works, as a reminder and a review, if we take a look at the right-hand part of the slide toward the middle, we have our thymine and adenine, our AT hydrogen bond. And specifically, the hydrogen bond here, I'm highlighting in yellow, is between the hydrogen atom that is bonded to a nitrogen here, so that's your positively polarized hydrogen, with a negatively polarized oxygen atom of the carbonyl. We come down to the next spot for hydrogen bonding. We have a nitrogen atom that is negatively polarized, that is engaging in a strong intermolecular interaction hydrogen bond with the hydrogen over here that's bonded to a nitrogen. And we come down and we can continue that network by looking at the same sort of theme occurring with guanine and cytosine, where there's actually 3 points of hydrogen bond interaction here, here, and here. And in each case, it's a strongly negatively polarized atom engaging in an intermolecular interaction with a positively polarized atom. And the two strands of DNA are referred to as being complementary to one another. And complementary means that the A is always hydrogen bonded to the T, G is always hydrogen bonded to the C. And so you can predict, if you know the sequence of one strand of DNA, you can predict that it is going to be hydrogen bonded to a strand that is complementary to it. And so what I mean here is, since we know that A always is complementary in hydrogen bonds with T and G with C, if we want to predict the second strand, so say we have a 5' to 3' DNA sequence that looks like this. Remember that the 5' is listed on the left, 3' is listed on the right. We can draw in the complementary strand that would hydrogen bond with to form a double helix. And it's always going to be that T teams up with A. So I'm just going in and listing through here all of my complementary nucleotides. So we're showing in each case, based on the top strand that's in blue, we can predict the exact sequence of the bottom strand because the pattern is always the same about who hydrogen bonds with whom, always A with T, G with C. The other thing to keep in mind is when you're looking at this partner strand, the orientation of the 5' and 3' ends are flipped. So it is always going to be the case that the second strand, the 5' portion, the free phosphate group is listed at the right end, and the free hydroxy group is listed at the left end in this complementary strand. And ultimately, this is the hydrogen bond pattern that enables DNA to assemble into its coiled structure. This coiled structure that I'm showing in the middle of the slide, the DNA double helix. So that double helix is driven by hydrogen bonding. And then the next layer of organization, which is sometimes overlooked but is important when we think about the function of DNA within cells, is that there is a tertiary structure. And the tertiary structure of DNA is formed by further interactions of multiple DNA double helices coiling together. And that's one way that DNA is packed compactly within a cell. And sometimes the DNA also engages in interaction with proteins to further tightly pack and coil it within a cell. So the DNA tertiary structure is going to be formed by interaction of multiple helices. So in the secondary structure, we're looking at one double helix with hydrogen bonding being the driving force of that. And now in the tertiary structure, we're looking at multiple helices coming together. A main example of that is supercoiling, which is how the DNA packs up into a tight little structure to minimize the amount of space it takes in a cell. And also by compacting that tight structure, it's thought that improves the stability of the DNA as well. So that is our 3 levels of organization of nucleic acids: primary, secondary, and tertiary. We're applying this primarily toward DNA in this particular class because one of the things we're going to be doing is manipulating DNA in order to create specific chemicals. I know there's a few steps between DNA and creating chemistry, creating small molecule chemicals, but we're going to be learning about that later. And in order to understand that, it's essential to grasp how DNA is behaving at the molecular level. So we will conclude with that for now.